And then maybe for those who don't know you already, I will give a very quick introduction. So Elisa Zamboni is a neuroscientist, I think mostly vision and kind of also body parts, looking at kind of topographical representations in the brain at high resolution. After a postdoc in, I think in Cambridge, was it, where I saw your name first time in Layer-fMRI papers, you went to York, and I think there you worked with Anirban Canale. But now just on the slide, I saw that he was not a senior author, so maybe he was a close collaborator, but not kind of the supervisor. But now back, you are back to Nottingham as faculty, and today Elisa is going to talk about high resolution 3 Tesla fMRI using Layer-fMRI CBV sequences to look at curvature domains in V4.
Yeah, thank you. So let me share the screen again. Yeah, thanks a lot for the invitation and for the introduction. So yes, the title is about mapping curvature domains. Haha. Give me a sec. Okay. Mapping curvature domains in human V4 using 3 Tesla and Layer-fMRI. However, the talk is going to take a bit of a longer turn before we get there. But we'll get to that. And this is so the reason why I thought to talk about some other aspects is also the fact that as Renzo has mentioned, I'm mostly interested in vision, and in vision as well as in fMRI, it's all about the scale. So the idea here being that if we're going out for a walk and we encounter something like this, a landscape like this, there's a lot of visual information that we can recover from this type of image. We can divide it into lower level visual features such as contrast, spatial frequency, color, orientation, or we can go all the way the other end of the scale, which might be something like, is there an object? Is there a face present? Is this image? Are there bodies? Or somewhere in between. So things like shapes and features or textures and contours, which are neither low levels, but not quite a category or an object that we can classify. And the idea here that really binds things together, for me at least, is the fact that when we talk about scale in the visual domain, we can also talk about scales in the way we try to understand the brain. So we can measure behavior and try to understand how we produce certain behaviors. We can look at several or a network of brain regions and put this together. So, or we can go the other extreme altogether. We can look at individual elements, so individual neurons, or how information from different neurons are collated together and merged into a slightly higher level of information. We can look at how this is organized into brain regions and how it's mapped in either a retinotopic or somatotopic kind of structure. And I'm sure all of you are familiar with these images because they are shamefully stolen from Renzo. The thing that really binds everything together is layers and columns. So this kind of sub-millimeter structure that can bring us to put together information that and gives us this kind of circuitry understanding of brain function. And my journey with dealing with layers and columns really started during my first postdoc in Cambridge, as Renzo mentioned, where we actually were interested in some more low-level visual features, or even something lower, in fact, because what we looked at was sensory adaptation. And sensory adaptation is a fundamental property of brain function that allows us to enhance our sensitivity to changes in the environment that is surrounding us, and is generally defined as the psychophysics microelectrode, which highlights the strict contingency between sensory, neural change, and perceptual outcomes. So this ability to link together what's happening out there in the world with what's changing in the brain and how we perceive the world itself. The way the sensory adaptation has historically been investigated was by choosing a specific visual features, let's say orientation, and then essentially presenting that kind of visual features over and over again over time. And this would be the so-called adaptation phase. And after a period of time, participants will be presented with a testing grating, usually vertically oriented. And the task of the participant would be to judge whether the orientation of the test grating is clockwise or anti-clockwise compared to vertical. And most studies really focus on this phase, so the testing phase, because it gives us information on how we process orientation in this case. However, in the design and in the project that we run as a collaboration also with University of Maastricht, we wanted to understand what happens in the brain during adaptation. So during this phase where we are constantly presented with the same visual input and visual information or sensory information over time. And why is this phenomenon so important? So we did that, as I mentioned, it was a collaboration with the University of Maastricht by using actually a 7 Tesla scanner this time, standard bold the gradient echo EPI. And the design that we used was the following. It was a block design, 2 conditions, a non-adaptation condition in which participants were presented with gratings that had different orientation at each point in time, and then an adaptation condition in which the same orientation was presented over time. Concomitantly, participants were performing also an orthogonal task at fixation, and this was to ensure attention. So they were presented with a stream of letters that was containing a target letter, in this case, the letter X. And participants essentially were supposed to monitor this stream of letters and count how many times they encountered the letter X, and then respond once prompted on the screen. So the hypothesis and that we wanted to test behind this design was the following. So we used a standard model, a very simplified model of the cortical structure and the circuitry that we know is present, such that we know that sensory information input reaches the cortex at the level of the middle. Sorry, I've just noticed that I've been moving my mouse around and it's not shown. So the level of the inputs reaches the cortex at the level of the middle layers, is then processed throughout the cortex, and then is moved forward or fed forward essentially at the level of the superficial layers, and then reaches the next step of processing throughout the cortex in the middle layer of the higher up area in the hierarchy of the visual cortex or the visual system. At the same time, feedback information is then mostly transferred throughout deeper layers of the cortex. So the hypothesis here was that if adaptation is essentially a mechanism through which recurring sensory input is monitored and fed forward in a sort of cascade throughout the visual cortex, what we would expect would be an enhanced connectivity between superficial layers of downward stream areas and middle layers of upward stream areas. However, we also usually have some kind of expectation of what the world is supposed to look like and behave. And other studies have shown that areas such as the interparietal sulcus or IPS is involved in this kind of expectations and predictions. So the alternative hypothesis, or the second hypothesis that we wanted to test, was to see if there was any kind of involvement of these expectations, and therefore whether we could see any kind of feedback connectivity between IPS and early visual cortex, so these kind of deeper connections in the circuitry. So how did we do that? First of all, we had to identify our regions of interest. So we used standard retinotopic mapping techniques. And so here we have our rotating wedges that generates these phase encoded waves of activity across the, so this is the occipital cortex. We then essentially flat out, so cut out the occipital pole and put it on a flat map. We then can use the retinotopic or visual field maps to identify each individual regions, so between V1 and V4 specifically, and then IPS as well. Once we've done this for each individual participants, we also obtain cortical layers for each of the regions that we were interested in. And this was done by first, obviously, manually adjust the segmentation, the gray matter segmentation across the occipital cortex. And then we used an equivolume technique algorithm to identify superficial middle and deeper layers of the cortex. And a kind of 3D representation is shown here. So once we had our layering for each of the individual regions of interest, we then performed functional connectivity maps, connectivity analysis. This cartoon here is just to remind you that in green we have our feed-forward connection, and in red we have our feedback connections. So if we start by looking at pathways involved in visual processing, here we can see that what we observed was an enhanced connectivity or feed-forward connectivity between superficial layers of V1 and middle layers of V2 during the adaptation conditions. So the results here of the connectivity is always a enhanced connectivity for the adaptation condition compared to the non-adaptation one. And so what we observed was these enhanced feed-forward connections between V1 and V2, and this also was repeated across the visual cortex, so between V1 and V3 and V1 and V4, which kind of suggests this, that effectively there is this incoming sensory input that is then fed forward in a kind of cascade throughout the visual network, plus some short-range feedback connectivity between V1 and V2. And as you remember, the second hypothesis that we wanted to test was to effectively look at the involvement of IPS. And what we observed in this case was only a kind of feedback connectivity between IPS and the deeper layers of V1. And this was really interesting because it kind of suggested that adaptation can be thought of as a constant updating of sensory information, which is then broadcast along the visual pathway and the visual cortex in a feed-forward fashion. But also there is this process integrates expectations from IPS through this feedback connection, and so this feedback mechanism. So what we interpreted this is that there are expectations, there are expectations, let's say, from IPS that are matched with the sensory input at the level of V1. And based on the match or mismatched of these information, the updated sensory input is then fed forward through the rest of the visual cortex. And this kind of mechanism can allow us to identify differences or changes in the surrounds. So when we finally finished this project, which was my first really work with laminar fMRI, I was really happy and enthusiastic. I was like, oh, I found my vocation. I figured out what I want to do, and the basic recipe, which is you find the gap in neuroscience, whichever pathway we weren't be able to tell apart before because the resolution wasn't there. We get the method running, you put your teeth into whatever Layer-fMRI method you have, you test all possible softwares that you find out there so that you get exactly what you want, and then you find the answer. Then you write up the paper until you find reviewer 2, which tells you actually you've used bold fMRI and that really doesn't mean anything. Now, this is kind of a joke, but what we're trying to say here is that we know that when we use bold fMRI, well, when we use fMRI in general, we're indirectly measuring brain function. And BOLD especially relies on a series of mechanisms. So it's blood oxygenation changes, which is changes in oxygenation, in blood volume, um, blood flow, and so it's a very complicated signal to understand, and especially we know that it provides a very blurry kind of maps of activation. So here is an example. This is data from our 7 Tesla study, and as you can see, we have the activation is actually blurring into or leaking into what should be CSF, which when you want to do a high resolution and study and try to understand how information is organized in the cortex is not very helpful. So I have a colleague that basically compares using bold fMRI to try to understand what's happening in a factory by looking at the sewer. So just because something is coming out of the factory, it kind of gives you the idea that something is happening in the factory itself, but you can't really tell apart which department is working or what's happening inside it. So in a less, I don't know, funny way to say it, and I'm sure we've seen this so many times, essentially what's happening is as soon as we have tissue activation, right, there is a cascade of all other events and mechanisms that take place, from oxygen consumption, increase in blood flow, which then generates changes in blood volume. And all of these have some level of effect to the signal that we measure. So which obviously affects the interpretability of what we are measuring, not to mention the resolution of the signal that we're looking at and the localization of it. So in the second project to try and that we run at this point, I was then in York. We wanted to try and find a method that was a bit more specific. And for this reason, we decided to turn on to using vascular space occupancy. And I'm sure that most people here know what we're talking about, but just very quickly. So if we assume a typical voxel to be a two-compartment structure, which is roughly 94% built of tissue and 6% of blood, we know that these two compartments have different T1 relaxation times. So in a vaso acquisition, we essentially provide a first inversion pulse. We then wait for these two compartments to relax back. And once we excite, and if we excite at exactly the point at which the blood signal is nulling out, then what we're measuring is the signal that is coming just from the tissue. And because during activation, what it's happening is that we have an increase in blood volume, so the vessels take up more space. We have a decrease of the amount of tissue within the same voxel, which then means that the signal that we are measuring is actually a decrease in the MR signal, which is then thought of as being proportional to changes in blood volume. And this is what we're actually using. And this is what we've used to run these curvature domains and mapping curvature domains in human V4. Now, curvature is yet a different type of visual feature, is not quite low level. So in curvature, you have information that is both about orientation, about to some extent about texture, and about background and foreground information as well. So it's not quite low level. It's somewhere in the between. It's not yet an object. It's just somewhere in between. And we have there has been lots of studies that have shown that curvature and contours are encoded at the level of V4, which is located in the ventral portion of the occipital cortex down here, which also the location itself represents a reason to use a kind of sequence that minimizes the effect of large vasculature, because most of the times around here we have one of the main arteries and veins and vasculature that is running, which already affects the signal that we're measuring from this region. So the idea here was that in optical imaging studies in macaques and as well as electrophysiology studies in macaques have shown this kind of curvature preferences of the curvature domains in area V4, which is shown here on the right. So we have these white and black blobs, which represents preferences for curvature versus straight lines. And as you can see, it's a very structured organization. And in this specific paper, they also show that there is this kind of map interacts with maps related to also preferences for colors and orientations. Now, in humans, there have been fMRI studies looking at curvature processing of curvature information in the ventral pathway. And they have observed curvature responses patches along the ventral stream, and some of these patches also overlapped with area V4. However, the kind of studies that were done, they all used bold fMRI, and the kind of resolution was not high enough to tease apart kind of domains level. So in our study, we invented the VASO sequence on a 3 Tesla scanner, so a 3 Tesla Siemens Prisma. The resolution that we actually ended up using was a 1.2 millimeter isotropic voxels with the TR for the combined images of 2.6 seconds. The design itself, so we still have to map individual retinotopic maps and individual areas V4 using retinotopic mapping. We had a total of 5 participants in this study. One of them was tested twice as a form of test-retest reliability, a few weeks apart between the 2 sessions. The stimuli that we used were very simple radio frequency patterns. So we had, in this case, for curvature, we used concentric circles, as shown down here, whereas for straight lines, we used concentric squares. In fact, the squares had rounded edges, and I might talk about this later on in a bit. The design itself was a simple block design with 30 seconds on, 30 seconds off, roughly. So for 30 seconds, participants were presented with concentric circles, and every half a second, the contrast of the radio frequency patterns was inversed to prevent any adaptation effects. Then they had 30 seconds of rest, and then another 30 seconds they were presented concentric squares, and so on. For roughly 15 minutes. So one single run was 15 minutes, and each participant performed up to 4 runs in one session. There was no active task. They were just looking at the screen and trying not to fall asleep. Okay, so because it was the first time that we implemented this type of acquisition on the scanner, we first had a look at the type of data and the data quality that we obtained. So as I'm sure you're all aware, for each TR, we obtained 2 different images, one with a vaso contrast and one with a bold contrast. And as you can see already from the vaso image, the type of gray matter, white matter contrast is quite high. Which is a good indication that the acquisition was working well. We then also looked at the TSNR values. So as expected, so the TSNR for the bold is in red, and the TSNR for vaso is shown in blue here on the graph on the side. So as we expected, the TSNR values for bold was much higher than for vaso. However, when we looked specifically at the region of interest, which are the plots shown here, we can see that even for vaso, we are working in the range of roughly value of 30, which are values that are comparable with some evidence reported at 70 as well. So we were quite happy with these results. And even when we then looked at a simple stimulus versus fixation contrast, so in this case, these maps show there's no difference. So it's a response to any stimulus that was shown on the screen. we can see that we have a reliable response. So both bold and vaso peak around roughly 10 seconds post-stimulus onset, and then they go back to baseline once the stimulus is offset as well. And in terms of the mapping onto the brain, we can see that it's quite consistent across the contrasts. So in general, the quality of the data and the sensitivity of it was quite good, and we were happy with it. Which then allowed us to move on and look at effectively whether we could see any kind of curvature domains in human V4. So as I mentioned, so this is the data for one participant. We have left and right hemisphere on each row. Here is to identify where the visual area V4 is located. And then we have the squares versus circles, or curvature versus straight lines maps for bold and for vaso. And as you can see here on the side, when we look at the vaso maps, we can start to see this kind of stripe pattern where we move from a red mapping, which is a preference for curvature, to a blue ones, which is a preference for straight lines. Now, these are only surface maps, so what we're doing is effectively projecting all the information onto the surface, pretty much as you would do with trying to identify visual field maps. So there's no information about depth in this case. And what these maps are showing is that effectively there seems to be more information or more structured information into the vaso maps compared to bold, which are more blurry. So this is the data for one participant, and these are for some of the others. And in general, we see that this contrast between the bold and vaso maps is persistent across all participants. So the vaso maps seem to provide more information. And on the right here, we have the same subject tested twice. And as essentially what we see is that we can reliably repeat this structural organization or dysfunctional organization across different scans as well, which again suggests that the methods that we're using is quite consistent. So despite the fact that we do see some organization, it's worth noting that we also saw a lot of variability across participants, which is definitely something that we want to look more into in the future. Now, obviously, I said that vaso maps seem to be more specific and have more information in the structure of these curvature domains, but this is very qualitative, and we try to put, I wouldn't say put a number, but we try to quantify this kind of sensitivity and specificity of these maps by computing a manual principal component analysis. So in this case, what we did was to extract these surface maps for each of the in each region and each hemisphere and each participant. And essentially, you can imagine kind of rotating this map and collapse it across the main components. And the idea here is if we expect a curvature domain to be functionally organized, we would expect the component to have a sort of sinusoidal shape. So an example for one participant and one region of interest is given here. So the more sinusoidal these components, the better or the higher structure in these curvature domains is present. And so here we have in blue the principal component that is extracted from the vaso map, and in red the principal component for the bold map. Excuse me. So as you can see, the vaso map seem to provide more information. Excuse me. Compared to bold. My voice is gone. Um, as a second, I'm really sorry about this.
Don't worry.
Um, as a second form of quantifying this specificity, we also followed some work from Alessandra by looking at an index of sensitivity, which we can identify as a response to any type of stimulus that we used, and also a measure of specificity, which can be thought of as an index of tuning to curvature. And so on the maps here, on the left-hand side, we have that we're plotting sensitivity along the x-axis against specificity along the y-axis. And so at the top we have bold, at the bottom we have vaso. And as you can see, and probably as expected, I would say, bold has a much wider range in terms of sensitivity, and this is why no is probably one of the reasons why bold is so widely used, right? Because it gives us a lot of signal. However, vaso definitely outranges bold in terms of its specificity. So there is a much wider spread of values along the y-axis for vaso compared to bold. So most of the information here is clustered around these ends, which again suggests that vaso provides a much higher specificity compared to bold. And again, this is in line with the results that were shown for 7 Tesla as well, which is very comforting. So up to now, I've only talked about surface maps. So what happens at the level of the cortex or through the cortical depth? Can we still see these curvature domains across cortical depth? Again, just as a reminder, in red we have a preference for curvature, and in blue we have preference for straight lines. So if we are to essentially move through our slab of acquisition, we can see that we have these blobs that start to emerge and follow through across from the surface down to the deeper areas of the cortex. A different way is that we can actually visualize this is to essentially extract the chunk of cortex located around the area of interest and then flatten it out into a disk, project our activation map onto this disk, and then it's much easier to actually see that these domains do run across the cortical depth. Now, this is a simple animation, but if we wanted to look at in a more static way, so again, you have the surface here, up here. We can then cut this disk along 2 of the main axes, and we can see that again, we have this curvature domains that are running through, that are running through the cortical depth as well. So this study effectively was the first time to show cortical curvature domains across the cortical depth in human before, which was really exciting. And the idea here is that hopefully we'll be able to carry on this work and using more complex stimuli as well, and look at the interplay between curvature and other visual features such as orientation and color as well. Now, hopefully I still have a bit of time, because so far we've talked a lot about space and different scales in processing and acquisition in terms of space, but we haven't really talked about time. And I'm sure, so this has been mostly driven by a collaboration with Manchester Metropolitan University, and I'm sure a lot of you have heard about this paper by now, and we might be also tired of hearing about it by now. But so when this came out, so this Diana paper came out, it seemed really big innovation, where they essentially looked at, or they showed, what they claim to show, correlation between spiking activity and this Diana measure that they had during electrical stimulation in mice. And so because at Manchester Metropolitan University we were already working on real-time acquisitions, we then tried to replicate this paper, as well as other groups did. So in this case, we used electrical stimulation of fingers. So we had, first of all, identified the representation of each individual fingers of the participants that we tested onto somatosensory cortex. And then we used a digitimer to check the input and output of the stimulation at the microsecond level. And the stimulation was essentially a 5 Hz stimulation to the index finger for up to 10 minutes. Now, when we first looked at the output, we thought, as well as other, groups, that we had been able to replicate these results, right? Because we had this kind of peak in roughly the correct location between 17 to 20 milliseconds post-stimulus onset. However, the same way as other people did, we also wanted to look at our data a bit further. And what we observed was that by looking at each coil element in our channels, we started to see these striping artifacts. And once we removed the information that was coming from these channels, from these coils, essentially we could see no longer this spike, this response was no longer available, which kind of suggested, as other people also suggested, that the results were kind of artifact driven. The one thing that we actually, while we weren't able to replicate this, we also thought it's still a very good technique. It's different. We shouldn't dismiss it, especially because we work with really quick events. And one of my PhD students was actually interested at that time in in binocular rivalry, essentially, or how we fuse information from the two eyes to obtain a coherent sense of vision. And we kind of stumbled upon this paper by Ravi in the late 90s, where essentially they used fast fMRI to look at activity or change or delays in whether it's possible to measure delays in response following delays in stimulus presentations. So what we replicated here was essentially we had a condition where we presented individuals with well, not fast, but flickering checkerboard, which were presented full screen. And then in a second condition, we started off by showing only half of the flickering checkerboard. So we stimulated only the left hemifield, which would elicit a response in our right hemisphere. And then 1 second after the flickering of the left hemifield, we then started showing the right hemifield as well, which then should result in a response in our left hemisphere. And in agreement with what Ravi showed, essentially when the stimulus was full screen, we also saw a response between the two hemispheres that was consistent across time. However, when we had this delayed stimulus onset, what we saw was an initial response of the right hemisphere, which peaked the first time when we actually would expected it to peak. However, at this point, we also had the second half of the stimulus turning on, which then started to elicit a response in the left hemisphere, but created this kind of shoulder and double peak in the right hemisphere as well. So, and this is essentially a plot of the same information. So we're looking at the phase delay, and we see that when the two stimuli are present on the screen at the same time, they match. Whereas when there is a delay in the stimulus presentation, also the response, the phase response and the peak, they kind of deviate from this line. And we found this very interesting because even though we would expect, for example, the vasculature to be somewhat independent in the two hemispheres, at this point, it looks like somehow the other hemisphere knows what's happening to the other part of the brain as well, and is affected by it. Now, we didn't run many controls on this. It was a very much of a one-off. And the reason why we did it was mostly because we wanted to use this kind of fast fMRI to look at binocular rivalry, as I mentioned. So in this case, participants were wearing glasses like this. And this is because we were then presenting different stimuli to each individual eye. So this is a representation of the task. In this case, we had the participants were shown this kind of concentric annulus to start off with to one of the eyes, which meant they were only seeing that specific annulus, concentric circles. After a short delay, we then presented a kind of radial gratings onto the other eye, which resulted in the suppression of the first image, and what they were then perceiving was only this kind of radial annulus. We then left the participants there for for 100 milliseconds or so, and after some delay, we then changed the contrast in a specific location on the image and to the eye that was currently suppressed. And the idea here is that that change in contrast then elicits this specific image to come back into awareness, so to say. So it kind of starts to break through such that over time, the stimulus that is the radial grating here kind of fades away and the concentric circles starts to come through. And the way in which this should happen is like what is defined as a traveling wave. So it's not an all on-off kind of mechanism, but it kind of gradually travels along the annulus. And then the participants were supposed to press a button when they saw this wave reach the bottom of the circle here. So what we did was essentially to use this fast fMRI technique. We identified the portion of V1 that was representing the annulus, or the portion of the annulus we were interested in. And then we looked at the trials in which this traveling wave was elicited and selected essentially or identified the voxels that were responding and plotted the response of the stimulus during this traveling wave time period. We then fitted and identified the phase. We took the phase of each individual response of the voxels as a measure of delay from stimulus onset, which would be the change in contrast here. And as you can see, we can kind of start to see a very specific organization of these traveling waves. So we have that the higher delay is effectively mapped, well, backwards in this case. So it would be from down here up. Oops, sorry. Up along the visual cortex. So again, these are only preliminary data, is only one subject for the time being, but it kind of again suggests that we shouldn't dismiss the use of fast fMRI because we can really use it to identify pathways and how visual information is encoded. And the longer term idea here is to try and use this information to see how we can disrupt perhaps this kind of traveling wave, or how it can be used to inform visual processing in individuals that suffer, for example, from amblyopia, which is this forced suppression from one of the two eyes. Yes, so I hope I didn't take too much time, but that was not too much about curvature, but some of it. And this is some of the people that have helped me throughout with all the studies that I've been talking about. Thank you.
Thank you.
Shall I stop sharing?
So we do have 12 minutes for questions for sure. And I see Lars switching on his video. Maybe he has some questions.
No, thank you. Nice talk.
Thanks.
No question right now.
Okay. And maybe I have questions then to start with. Maybe a practical question for the curvature study at 3T. Like we specifically temporoventral, we tried, also kind of hippocampus we tried, and it's so hard. And it's like not the laser that makes it hard, but the readout. And 3D-PI helps a little, but only so much. So my question is kind of, you're doing it at 3T. Was that more a decision of kind of that was was available, or was that a decision based on kind of ventral is just hard at 70, and that's why you expected to have an advantage at 3T?
Yeah, so well, the short story was that was available at the time. So this was the actual data collection was done right after. Well, I was in York, so I didn't have any availability for any 7T time, scanning time. And also, slight disclaimer, this work was not supposed to be my work. So I had a colleague that was actually involved in the grant, but they couldn't do anything with it. So we kind of went and ran with it. But in general, yeah. So the short story is the 3T was what was available. I also, yeah, definitely it helps in the sense that a 7 Tesla, we know we did try as well when we were in Cambridge to do some hippocampus work as well, and it was just, yeah, it's really, really tough. Um, but I think in general, just the idea of opening up the possibility of doing high resolution at 3T, it's really important. I think it's really important because it's not so easy to get access to ultra high field scanners, even if you're working in a center that has it.
Okay, noted. Maybe also about this study. So you showed the vaso and the bolt, and like very often when I look at layers, kind of the pile veins are just very hard to get rid of in bold. Looking at curvature domains, you have more flexibility. Um, or specifically looking at kind of columnar structures, topography. Um, Usually I'm in kind of my data, I see that kind of vaso can be um, slightly more delineated and crisp compared to bold, but for you it's really like a different world. So I know V4 is weird and doesn't it have this Venus eclipse or what is your explanation? It has more vein problems than other areas, does it?
Yes, so that's one something that I thought I mentioned. So it has this Venus eclipse. So in bold, it's even in a normal study, even with much larger or lower resolutions, it's really blurry. And even doing some most of the times, even just doing simple retinotopic mapping, it can be really tricky. So identifying borders of area V4, there's so much variability across individuals in terms of retinotopic mapping, and it's really hard to determine whether that's just functionally driven or it's due to the fact that the bold signal is so bad because of the of the vasculature that runs around it. Um, so I didn't mention we did acquire also SWI images in this project. So we kind of used that information as well to make sure that we kind of didn't hit the voxels that were either on or near really near the main vasculature. So we removed those. Yeah.
Cool. If nobody else has questions,
I have questions.
if go ahead. I have plenty. Yeah, go ahead.
Okay, so with this Venus eclipse stuff, do you mean you didn't say, but I'm curious. Like, do you say the vaso contrast looks better there, even though there's Venus eclipse, or is it just independent for you?
So in my view from the data that I've had, it looks vaso looks better in general. So the bold is really blurry, even just the images. Um, but then to make sure that we kind of control both contrasts equally, so to say, then we use these SWI maps to remove those voxels for both images, if it makes sense.
I see. Yeah, I asked this because I was like discussing this with another colleague recently, and I thought that actually vaso wouldn't help there because in vaso with the blood nulling, you get rid of the arterial blood, right? So, but this effect that you are talking about, this venous eclipse, that is due to a large subdural vessel vein,
Yeah.
ex, kind of
Yeah.
creating a large susceptibility distortion around it.
I know
So
I
I
was
think
quite
actually there you don't expect vaso to help you. right?
I know. I was quite surprised. I just
Also for Renzo, maybe I'm making this comment.
Well, it does blood nodding. Like the T2 star should still be the same in bold and vaso. So I think the bold contamination from that large vein shouldn't be there, but the overall dephasing effect, I would expect that it's still there. So if that's what you mean, right? That you just don't have signal
Yeah.
around those large veins, no matter what. You don't have bold contamination, but also you might have false negatives, maybe in vaso. Maybe about these domains.
Okay.
Sorry, go ahead.
Sorry. And my last question. So I saw that you use the LN2 multilaterate and patch flatten in Laney. I'm quite happy to see that you are one of the few users that adopted it, and without me being involved. So I'm curious about your user experience. Like, how easy was it to run this all like flattening pipeline?
so I think first of all the video tutorials that you've made are amazing. So I think I've known them by heart by now by following those. In general, it was I think it was fairly simple to follow the steps. I had some issues because obviously, as you do say in the papers and in the tutorials as well, these are meant for much higher resolutions than what I had. So I had to upsample the data quite a lot, which then kind of meant they got just blurrier and blurrier. So that's just the kind of technicality that made it hard. But from a point of view of using the tools itself, I thought that yeah, that was really, really easy.
Okay, cool. Good to hear.
And the documentation is really good. So
Okay, yeah, good to hear. Good to hear.
thank
Thanks.
you.
Hassan?
Yeah, thank you. That was a great talk. I have a very brief question about the TSNR values that you showed for bold and vaso in V4. If I'm not mistaken, in V4 for vaso, the average value, like the peak was around 30. And I was wondering whether you have done any sort of denoising or like, was it like Nordic applied or something like that?
No, so this didn't have any kind of denoising applied. We did consider using Nordic, but by that point the data was already collected. So I think it's possible to simulate the noise that you would expect and then use that retrospectively, but we didn't do it. So it's possible that by using that kind of techniques, it would even get better. So, but this data didn't have any kind of denoising applied to it. So the TSNR values are essentially how they come out of the scanner.
Okay, that was nice. Thanks.
Thank you.
Can I have a question?
Oh, yes.
Yeah. Thank you for them. I'm so happy to see the study has been done using 3 line for Layer-fMRI. And I have one question about the spatial resolution. I thought in your current study, the seems like 1.2 millimeters. So do you have the chance to have more higher spatial resolution, like 1 millimeter or 0.9 millimeter? Because in my experience, it's not so easy to see the layer activation in sensory cortex because the gray matter is very thick, like 1.5 and no more
Yeah.
than 2 millimeters. So do you have any experience about go further high resolution?
So we don't at the moment. When we were towards the end of, so just before I left York, we were testing it for 0.8 millimeter, but we essentially didn't carry on with it. I think we only collected a run and it was very noisy as well. So we didn't really put more effort into trying to develop that high resolution. It would definitely be beneficial because even in visual cortex 1.2, it's really stretching it if you want to do multiple layers, right? So the idea, so at the moment, what we're trying to do is work onto a grant proposal, and then hopefully that will kind of allow us to look at higher resolution as well. I agree, it's not what we would necessarily define, I think, as high resolution. nowadays. Yeah.
Okay, thank you.
So with these resolutions, you see that, or like it's hard for me to judge to see how to get a feel how big these curvature domains are in practice. Like I believe you showed the entire V4,
Yeah.
and they were like maybe 6 to 8 different curvature domains. In the monkey data that you showed, it was kind of encapsulated with those veins, so it's not clear if that's entire V4. Maybe it looked a bit kind of more than that, but also kind of stripy. But in these kind of animations that you did to quantify the selectivity, you had kind of 3 bumps in each direction. So is it my perception, or is it true that you kind of just had a maybe 2 handful of curvature domains in the entire V4? Is that expected? And they are kind of then 1 to 2 millimeters thick, or how is it? Yeah.
Yeah, so in monkeys, there's definitely a higher rate, let's say, a higher rate of changes. We didn't quantify, so in the maps that I've showed, we didn't actually quantify this kind of rate of changes. Mostly because it's really not so the thing that was quite striking was that it's hardly any consistency across participants, which then kind of makes it much harder to quantify as well. And for some of the maps, it's more clear stripes, which is what the example that I've showed with the only 3 bumps, whereas for others, it's really like blobby. The one thing that I would want to look more, which the higher resolution would help as well, is try and understand whether it's just a function of the way we are projecting all the information onto the surface, whether it's just blurring things, or if essentially why is there so much variability across participants as well. So that's something that I haven't done before, but I think it would be really useful and interesting to understand. I have to be honest, I don't have a specific prediction or expectations of what I would expect to see in humans. And I don't know how, there's a whole debate also once we get higher than area V3 on how much overlap there is between humans and monkeys just in the, right, what we define as V4 or V ventral V4 or V4H, or so that also is a whole level of debate, which makes interpretation much harder. But I think modulating the stimuli more would help tease apart this kind of organization more.
Yeah. Makes sense. Thanks.
Thank you.
I know we're a bit over time, but I do have maybe 2 burning questions left for this Diana stuff. Like first of all, it's quite impressive how many kind of things you do, and they're all kind of really high level, but also you need to go so deep to in the methods to really make it work. And that's
I think you know whose fault that is.
He's not joining today, is he? No. For the Diana things, you see these artifacts in some coils only. So you say those are stripes, and are those images then, or what causes this artifact? And are those the coils that are close to the motor cortex, or
That's
why
a
is the
whole
artifact at that time point then?
that's a whole mystery. So they are not always the same coils elements. So different people will show it in different places. Some people didn't show anything at all, but then we couldn't even see the response either. And I think the most striking things was at some point we had 8 participants in the scanner, and I was sitting next to them. The stimulation was on my digits, on my fingers, and we still saw the signal and this effects in the coils. So that was the kind of ultimate test of this is an artifact. But we tried to remove, we thought maybe there's some conductivity with the spinal coil or something. We tried to remove that. We couldn't really tease apart what's generating it. And so we kind of abandoned it in that in terms of it's definitely something technical, but we couldn't really get to the bottom of what was driving it, if it makes sense.
The left-right hemisphere thing is really cool. I know Peter Banettini always likes it and wants to kind of wants us to continue working on it. If I understood it correctly, kind of there's this very kind of few millisecond apart delay between the left and right hemispheres, but then in the HRF you have these several second delay, which might be very interesting based on kind of your hypotheses of kind of how the transcallosal inhibition or connectivity might be responsible for that this comes in different layers, right? So are you planning to do this at layer resolutions or?
I would love to. It needs time and money and a student that is willing to do that. But yeah, so the whole project was started with that on the long term, getting there eventually. But yeah, we are at the very like first step kind of. But it would, yeah, I agree, it would be really cool. And so even just the simple flickering, left-right flickering. So I think what we've been trying to tease apart is also if we only use one hemisphere, hemifield, and rather than the two hemifields, just the top and bottom are within the same, and look at what happens there, which is kind of similar to this traveling wave idea, but at least you're restricted on only one hemisphere essentially. But yeah, it's yeah, I'm sure we're all on the same kind of boat when it comes to accessing scanner and scanner time, and it's hard. But yeah.
All right, I don't have any further questions, and we are 6 minutes past. So if nobody else has any questions, thank you a lot.
Thank you very much.
On this kind of nice overview. Thanks a lot, and I hope you will have all the 70s time that you need in the near future. Yeah.
Let's see. Thanks. You take care, guys. Nice to
Bye.
see you.